All right, so for this next one is translation. So we already did transcription. And remember, transcription had 3 steps: initiation, elongation, and termination. And so what's messed with your head? Translation has initiation, elongation, and termination. Same steps, names, quite a bit different on how they work. So when you're looking at tests, when you're talking about it, be sure that you pay attention. Am I talking about transcription or translation? All right, so let's look at translation. Again, purpose. This is something that's going to be happening outside in the cytoplasm at the ribosomes. What you need to know is that we're now taking the messenger RNA that comes from the nucleus. Again, the exons with the guanine cap and the poly A tail. And what we're going to do then is we're going to take that and we're going to read that with what we call transfer RNA, which is going to bring in the proper amino acids to build the growing polypeptide or protein. Okay, so again, if you look very carefully, here we have the messenger RNA. Again, this came from the nucleus. This came from transcription. Here then we have a transfer RNA molecule. So the messenger RNA would have a group of 3, a triplet called a codon. The transfer RNA would have a triplet called an anticodon. And then this structure, the transfer RNA, will be bringing in whatever the correct amino acid is. So CCG is the anticodons. If you go to the table back and look at GGC, that would code for glycine. So that messenger RNA table, GGC, would actually code for glycine. Okay, so that's pretty much what we're looking at. And so what we're going to do is go through these steps. It's going to be kind of one of probably the longer videos, a little more complicated. So here's what you need to know. Transfer RNA is a single strand, just like messenger RNA. But it is bent into a 3-dimensional shape. And so what is important is a couple of different spots on it. One is the anticodon somewhere in the middle of this single strand. And then the other important part will be the 3' end, and that's where the amino acid is going to be attached. So here is the anticodon somewhere in the middle. This is all this twisted areas here. And then somewhere on the other end is the 3' end where the actual amino acid will attach to. Okay, what you need to know is amino acid transfer RNA synthetase. This is an enzyme that actually is floating around in your body. And what it's doing is it is trying to figure out, okay, what is the anticodons on this thing and what is the proper amino acid to attach it to. And so then it catalyzes and does that. So these amino acids are actually coming out of your diet. So when you eat proteins, it breaks it down to amino acids. This enzyme will pick that amino acid up, and then it'll pick up a transfer RNA and hook the correct ones together. All right. So let's talk about how this works. Okay, so there are ribosomes, and let's talk about the ribosomes. It turns out there's 2 subunits, and fancy names. There's a small subunit, which is called the small subunit, and then there's a large subunit, which is called the large subunit. So life's not hard. Now they're really separated, and they come together during protein synthesis. In the large subunit, there's actually 3 different sites. There's the one in the middle called the P site, which is the peptidyl transfer RNA site. What I want you to know, that is where the growing protein is happening. That's where the growing polypeptide is. Okay, the A site is the amino acid transfer RNA binding site, and this is where almost all the new transfer RNAs with their amino acids show up. All right, the E site is a newer site. It's in the last couple of editions, but it's called the exit site. That's where the transfer RNA gets shipped off to when it has lost its amino acid, and then it exits obviously back into the cytoplasm. Okay, so that's what you need to know for ribosomes. All right, now this is where it gets ugly. Okay, so there are going to be 16 steps. I'm going to go through the steps real quickly. What I want you to know, I will never ask you step 7. The numbers are mine, only mine. But I think this concept is kind of easier to get if you walk through it as a process. So again, I do not, the numbers mean nothing. I won't ask on a test what is step 14, because actually, to be honest with you, depending on how I do it, there's anywhere from 14 to 18 steps. So I usually don't type them out. I had to type them out for the recording. So here's what we want you to understand. Okay, so here's how it works. We'll start with messenger RNA will actually attach to the small subunit. And then you have that start codon. And it comes in. So it has the AUG on it. So a UAC attaches the anticodons, which brings in MET. Okay, so there you go. There's your messenger RNA, AUG. Okay, we're good. So think about it. So that's there. Then what happens when that's sitting there, the large subunit comes over, and the first transfer RNA with its corresponding amino acid slides into the P site. Okay. That is initiation. Elongation now. What happens is the second transfer RNA with its corresponding amino acid slides into the A site. So now you have 2 amino acids sitting next to each other. Then what you do is a peptide bond forms between the 2 amino acids. Then you get something called translocation. So once that bond forms, the first transfer RNA will actually release its amino acid. It'll slide into the E site. The transfer RNA that's in the A site will slide over into the P site, and it's holding the growing peptide bond. So everything's pretty much shifting. That's called translocation. Okay, so continuing elongation, then the third transfer RNA with its corresponding amino acid enters the A site. Another peptide bond forms, another translocation. All right, the fourth transfer RNA enters the A site, another peptide bond translocation. And you just continue this over and over and over again. So again, what I want you to kind of key in on and kind of read this through and kind of think about it, but the first transfer RNA with its amino acid, the AUG and the methionine actually slides into the P site. So the first one slides into the P site. That's when the large subunit comes in. Every other transfer RNA with its amino acid will go into the A site. Okay, 1, 2, 3, 4, no matter how long it gets, 200, whatever length this protein is, going to continue to do that. Goes over and over and over. Okay, and then translocation is everything shifting. So the transfer RNA in the P site goes to the E site. Transfer RNA in the A site moves to the P site, and that again is holding, is going to be holding the growing polypeptide, and then again the next one will enter the A site. So it's happening over and over and over again. Okay, until you get to the termination, which is one of the stop codons. When one of the stop codons go into enter into the A site, instead of bringing in another transfer RNA with an amino acid, it brings in what we call a release factor. And that release factor goes in and actually kind of blows up everything. So the polypeptide now is free, goes to where it's going to go, either the Golgi apparatus, somewhere in the rough ER, whatever is going, cytosol, wherever it's going. And then the messenger RNA breaks free, and actually the small and large subunits break apart. So those are the 16 steps. I kind of typed them out. Hopefully you can go over those and see what happens. And then we have these signal peptides, and these signal peptides are basically kind of telling where are we supposed to be. And so you can see the ER lumen. ER stands for endoplasmic reticulum. You can see the ribosomes kind of attaching themselves there. And here we're building these polypeptides, which are going in the inside of the ER. Okay, so this is kind of newer stuff. This is signal peptides are basically kind of controlling where these peptides are actually going, these polypeptides. All right, what you also need to know is polyribosomes. That's a group of ribosomes together. And what we want you to realize is that you don't build all this, send it through a single ribosome, and then waste it and detach it. That seems like a lot of work for nothing. So what will happen is a messenger RNA molecule will be read by multiple ribosomes, polyribosomes. And what they find is the front end of the mRNA will enter a ribosome. And when it comes out the other side, it can go into another ribosome even before the rest of the mRNA is read by the first ribosome. It's kind of like if we're reading a book and somebody's reading pages and they rip the pages and then handed it to you. So you're reading the first page of chapter 1 while the person is moving on to chapter 2. So that way you can actually reuse that messenger RNA over and over again, which is pretty smart. And this is kind of trying to explain it all in one big happy picture. So I don't know if this helps or hurts, but a good thing that you should be able to do is maybe look at this picture and kind of walk yourself through it. And can you explain all the stuff that's going on here? And if you can, then you're ready for the exam, at least as part of the exam. So we'll stop it.